Okay. Okay, we start. Hello, everybody. Hello, everybody who are on the web also. So it's a great pleasure for the Labo Mixed today to welcome Professor Jan E and Dr. Gillian Lang from the University of Colorado. And Jan has kindly accepted to give us a talk on her favorite topics. And Jan is a very famous plant pathologist, and I have to read my note because you have so. She's a plant pathologist, and she is Associate Dean for Research in the College of Agricultural Science. She is also Distinguished Professor at Colorado University, past President of the American Phytopathological Society, and so on and so on. And finally, she is also President of the International Society of Plant Pathology, and this is the reason why she is here in Lyon, because we have to work together on the next meeting of ICPP. And finally, she is also a member of the National Academy of Science in the US. So one of her scientific challenges is to understand at the molecular level the interaction between plants, microbe, and also environment. So please, thanks, Jan, and we are listening to you.
Thank you. Well, thank you, Natalie, for the introduction, and thank you for all coming today. So, as Natalie said, we're here because the International Congress of Plant Pathology will be hosted in Lyon, and we're really excited about the possibilities of bringing plant pathologists from all over the world to Lyon to visit you and take advantage of your wonderful host relationships. So today I'd like to talk to you a little bit about some of the work that's been going on in my lab. This has been a side project of the lab for a number of years, but you'll see where we are headed, and you'll see hopefully the importance of thinking about the interactions between plants, pathogens, and the environment, in this case, high temperatures. So I don't need to tell you that warming in the ecosystem is unequivocal. It's happening. The International Panel on Climate Change has said that for the major crops, such as wheat, rice, and maize, we will see depletions in yields for most of those, and that this is happening at a faster rate than we anticipated in the past. So we need to start thinking about how are we going to adapt crops? How are we going to manage the interactions between crops and the emergence of plant pathogens on those crops? So this is particularly important to people like Gillian and I, who come from Colorado in the middle of the country in the Rocky Mountain region. We've been smashing records in high temperatures, which, you know, for the mountain region, 100 degrees is painful. Even if it's dry, it's painful. And this, in fact, has been happening and been causing more and more problems. So that, in fact, is my property burning. My barn, in fact, is the big blow up there in 2010. And that was sort of the beginning. We started to see a lot of wildfires in Colorado. And in fact, in 2020, 2 years ago, we had bought a new cabin higher in elevation, and Colorado experienced 3 of the largest wildfires the state had ever had in one summer. And so that cabin is now that pile of mess. So one moral of the story is don't buy property where I buy property. The second moral of the story is we're in trouble, and we need to be thinking about how are we going to manage the high temperatures and the fires in the future. For plant diseases, we know, or for plants, or for crop plants, we know that we're already seeing effects of high temperature, and that even a 1 degree change can affect the yields negatively. And already we've seen that in many parts of the world. And in fact, I'll talk to you mainly about rice today because that's kind of the crop of my choice. Rice is not only negatively impacted by increased night temperatures, that's the most important time of day for rice development. And so we've observed increasing rice temperatures of over 1 degree internationally, and this has been happening over the past 50 years. So we're already measuring effects on yield. And not only are we measuring effects on yield, decreases in yield due to the high temperatures, but with rice, we're seeing increases in the release of greenhouse gases as the temperatures increase. So it's in fact, a double whammy. So we've been wondering for a long time in our group, how will increasing temperatures impact plant disease? And there are a lot of models out there. And a lot of people have been modeling this for the past 10, 15, 20 years of what might happen and what should we do to manage it. A lot of predictive models, but until very recently, there were lots of models and hypotheses, but we really didn't have any empirical data to actually show what would happen if we had increasing temperatures and increasing, what would happen to plant disease. So a lot of people have started to study this now, and this is a very recent paper that just sort of sets the stage for this, and that is there are many things that can happen. We can have not only a shift in the host range, so as high temperatures increase, the host population shifts, but also new pathogens can emerge in those areas where that host has not been present before, or in fact, as this paper showed, you can get a shift in the pathogen levels with increasing droughts, with increasing temperatures, and shifts in the host range. Now, I once had an ecologist say to me, "Oh, Jan, you agriculturalists will figure this out. It's not a big deal. You just grow corn further north in the United States and you don't worry about it." But think about it. That corn was not adapted to the soils of northern US or to other conditions in the northern US. So you don't just move a crop north. You have to think about adapting that crop, not just only for the climate change differences, but also for the conditions that it's going to face and the pathogens that are there and the emergence of new pathogens. So today I'm going to talk to you about rice and one of my favorite pathogens on rice. So this is bacterial blight disease of rice. It's caused by Xanthomonas oryzae, pathovar oryzae. And it is the most important bacterial disease of rice worldwide. It's mainly found in the tropical regions of the world. So for many plant diseases, we have seen that disease pressure increases with increasing temperatures. So for example, with bacterial blight of rice, you can see that at 35 degrees days and 29 nights, which we choose as our high temperature, and the rationale for these particular temperature ranges is that 35 degrees day, 29 degrees night is the temperature, average temperature for the hot season in the Philippines, where I do a lot of my work, and the 29 day, 21 night is the average temperature for the cool season. So not only we, but many others over the history have observed that we have high levels of disease in rice in the hot season, especially if it's associated with high humidity, because this disease likes high humidity. And we have much less disease in the cool, dry season. And we can repeat that in the greenhouses and growth chambers. So this is a rice variety with no resistance to the pathogen in it. And you can see that at high temperatures, the lesions, which is our measurement of disease, are much longer. They're twice as long as in the low temperatures. So this is a real issue because we're moving towards higher temperatures, and we haven't really adapted our crops to that. So if we think about it, the predicted effects of increased temperatures on disease could either affect the host or the pathogen. And if we look first at the pathogen side of things, we could ask the question, is the increase in temperature at high temperatures related to increased multiplication of the pathogen in the host? And you might expect that the bacteria will grow faster or multiply faster and will move faster in the plants. And in fact, if we measure the lesions, so again, we have shorter lesions at low temperatures than at high temperatures. And if we look at the movement of the pathogen in the plant, the population of bacteria spreading down the leaf, which is the way this pathogen moves, it's a vascular pathogen. And you'll see that, of course, it can move further, faster at high temperatures than at low temperatures. But if you add those populations together, the bacterial numbers in these distal sections are not hugely different than, so the amount of bacteria in the leaf is not hugely different than in the high versus the low temperature. So we would argue that it's probably not going to be so much the numbers of bacteria. And in fact, as for most plant pathogenic bacteria, this bacteria doesn't really like to grow in high temperatures. So in culture, if we put the bacteria in culture over time and look at the multiplication rates, this is a bacterial growth curve, you can see that at high temperatures, it essentially stops. And that's the high temperature that we use in our studies. Now, we all know that plant leaves transpire. And so we know that the leaf temperature may not reflect the ambient temperature of the environment, and that is in fact true. And we've measured that. So if you look at the infrared data, you can see that the average temperature across the leaf is about 2 to 3 degrees less than the ambient temperature. But still, that's relatively high for our pathogen. And that, of course, this is external leaf surface, and it'll be a little cooler on the inside. But still, we don't think that the problem with the bacteria causing more disease at high temperatures is related to its better adaptation at high temperature. This pathogen is not adapted to high temperatures. So we then turned to looking at why we're getting these diseases at high temperature relative to low temperature. Somehow I've got some slides mixed up in here. Oh, and then we asked the question of what is the influence of high temperatures on disease resistance? So one of the things that's really important for controlling bacterial blight of rice is breeding for disease resistance. But what we've never really taken into account historically is the changing environment of when we deploy those plants. So if we have heat stress, our breeders are working hard to bring in heat tolerance. And we have pathogen attack, and we work to bring in disease resistance. But what we don't know is when we have those combined stresses, what is the effect? So when we ask that question of bacterial blight of rice, and the major source of control for this disease is single gene resistance, which the breeders have worked hard, and we have something like about 30 resistance genes, which are helping us to control this disease worldwide. But you don't have to be a plant pathologist to see that sometimes those genes break down, and they break down dramatically. When they break down dramatically. So what happens if we impose a high temperature on that? And it's quite interesting because most of the bacterial blight resistance genes that we've looked at break down at high temperatures. So the disease is much greater, and the gene is relatively ineffective, as is the case for XA4, which is one of the most widely deployed resistance genes for bacterial blight in the world. It essentially breaks down at high temperature. But there's an exception to that, and that's what we twigged on, and that's what we got very interested in, is this resistance gene XA7, which has the exact opposite effect. It's a very good resistance gene, but it's even better at high temperatures. And that's very repeatable in multiple labs and in multiple experiments. So why is XA7 more resistant at high temperatures? And why is rice more susceptible to disease at relatively high temperatures, even with other resistance genes? So we also did this to make sure we were seeing what we thought we were seeing. We did this using area under the disease progress curve, which is a more accurate way of measuring disease because you're measuring disease development over time. And again, XA7 is much more effective at high temperature relative to low temperature. So I need to tell you a little bit about bacterial blight and XA7. So Xanthomonas oryzae, the bacterial blight pathogen, depends on these effectors called TAL effectors. So TAL effectors are transcriptional activator-like effectors. In fact, they are transcription factors. We know that now. We named them before that. And they are major virulence factors in Xanthomonas oryzae. And one bacterial cell can have maybe 5 up to 25 TAL effectors. But there are certain TAL effectors that are more important than other TAL effectors. So they're not all created equal. Some TAL effectors are very important virulence factors. Others contribute minimally to virulence. AVER-XA7, the TAL effector that corresponds to the XA7 gene, is a major virulence factor. And if you knock it out in the bacteria, that bacteria is only weakly virulent. So it's a very important virulence factor. And it's been targeted by the XA7 resistance gene. So how do the TAL effectors function? Well, in fact, the bacteria injects the TAL effector into the plant cell using a type 3 secretion system. And then once that protein, the TAL effector protein, is in the in the plant cell, it is transported into the nucleus where it binds to promoter regions in the nucleus in very specific places. And the sequence of the TAL effector dictates where it binds in the nucleus. And then it activates the transcription of a host plant gene. So a bacterial effector gets into the plant cell, moves into the nucleus, and activates expression of a host gene, which is really exciting and interesting. That spot where it binds is called an effector binding element. And it's similar to a cis-regulatory element, but they are very specific sequences in the promoter. So just to give you a little idea of how this works, so what happens is the bacterial TAL effector comes in, binds to the promoter, and activates the expression of a gene called a susceptibility gene. Now we know that some of these genes are sweet genes, which are sugar transporters. And the hypothesis is that the bacteria secretes sugar out of the plant secretes sugar out of its cell and into the extracellular space where the bacteria can use it to grow. So essentially the bacteria is saying, feed me, feed me, feed me, right? And it's taking over the plant cell in order to do that. But for resistance, there are two mechanisms of resistance, actually more, but two that we focus on. One is where the TAL effector binds to the promoter of what we call an executor gene or a resistance gene. And this gene will, in fact, trigger cell death, plant cell death, or resistance. It turns on a lot of downstream genes, which are, in fact, involved in stopping the pathogen from multiplying in the host. And I'm going to show you in a minute that XA7 is one of these genes. However, the story is more complicated because for some TAL effector elements, what happens is the resistance is when they cannot bind to the promoter because it's been mutated. And so the susceptibility gene is not activated. So you have resistance because you don't have activation of susceptibility. So that's the mechanism that many resistance genes use. Now, I'll tell you that AVR-XA7, the tau effector that interacts with XA7, binds to a sweet gene and activates sugar secretion and feeds the bacteria. But it also, we now know, because just recently some colleagues of ours in a massive effort, because it was really, for whatever reason, a very difficult gene to identify and clone, 2 different groups have cloned the XA7 gene. And we now know that it's an executor gene. As I showed you, it's a resistance gene. And that in the promoter, there is an effector binding element that is very similar to the effector binding element in the sweet gene that the effector also activates. So this story is a little more complicated than some of the other tau effector sweet gene activation, executor gene activation stories, but it's actually quite interesting. So the hypothesis that they put forward in this paper is that the tau effector prefers to bind to the sweet gene and activate disease, but the XA7 gene can compete for binding of that tau effector, and when you get competition for binding for it, once it's activated, it sets off cell death and resistance. So the reason that's important to us is now we're trying to think of, well, how is it that AVR-XA7 and AVR-XA7 interactions are more effective in resistance at high temperature than low temperature? I'm not going to give you the answer because we don't know it, but you can see what the model of where we're starting to think of how is temperature impacting these interactions here. Now, it is very interesting that XA7, it turns out, is regulated by high temperature. So it's, first of all, you have to have the effector gene. The bacterial effector gene has to be present for the plant resistance gene to be turned on. That's a given. So it's only activated at the normal temperatures if you have, so these are duplicate samples here, if you have the effector gene present, AVR-XA7. But it's super activated at high temperature. So is it competing for the sweet promoter region? Is that the reason that it's more effective at high temperature? We don't know. It's odd because if you think about the dynamics of protein DNA interactions, you would anticipate that higher temperatures would make that less effective, the binding. And so you would have less binding to the promoter. But maybe there's something about the XA7 EBE, the promoter in front of the XA7 gene, that makes it more available for binding to the effector. These are questions that we don't know, but we would love to have the answers to. So I guess the question that we'll be addressing into the near future, now that we have the XA7 gene cloned by this other group, is how the regulation differs at high versus low temperatures. But we have looked at what's happening in the host plant at these 2 different temperature regimes. And you can imagine there could be many effects on the host plant, some of which would be altered plant architecture, which would impact the pathogen's ability to get in or to live on or in the plant, or changes in function of effective resistances, which is what we'll be targeting, and also different increased temperature responses. So a graduate student several years back did a meta-analysis of all the literature out there in rice exposed to individual stresses. This is not combined stresses. And asked, were there genes that overlapped between biotic stresses and abiotic stresses? And the answer he came up with is that there are indeed gene groups that are regulated differentially at abiotic and disease stresses, the common genes in an individual stress situation that change. So for example, we've known for a long time that in disease responses, you see downregulation of photosynthesis. We've also known in disease responses, as well as in salt stress or drought stress, that you have upregulation of ABA responses. So the question for us became, if these things are happening at individual temperatures, what changes in the pathways happen when you combine the stresses? So Stephen and Hongxia, who was a visiting scientist in the lab, designed a transcriptome analysis to begin to address this question. And the two questions that we were addressing were, why are plants more susceptible to bacterial blight at high temperatures? And then why are some resistance genes like XA7 more effective? So using a transcriptomes approach, we asked this question. I'm going to summarize a lot of data in just a few slides and try to make it clear. So this is a little bit different way of looking at a transcriptome analysis. So I'll walk you through it because I like it because for simple minds, it's very simple and I can understand it better. So it's called a kernel density plot. And what Stephen has done is he's plotted the log fold data of high relative to low temperature for different groups of genes. And this particular set of genes, and the ones that we're going to focus on, are the ABA biosynthetic and responsive genes. So the ABA pathway, abscisic acid biosynthetic pathway. So what you're looking at, if you look to the left of the 0, are the genes that are downregulated at high relative to low temperature. And on the right, they're upregulated at high relative to low temperature. So you can see here in the mock treated, which was water infiltrated leaves, that we have an upregulation of the ABA biosynthetic and downstream genes at high relative to low temperatures. So what happens if we add in, well, first of all, why are we interested in abscisic acid and why did we focus on it? We looked at many other things, and I'll show you a little bit of that, but abscisic acid is known to be an abiotic stress response. So drought stress, heat stress, it's known to be upregulated and to help in the adaptation to those stresses. It's also known to be antagonistic to the salicylic acid pathway in many systems. So in rice, it's been shown to be antagonistic to salicylic acid. And salicylic acid, for those of you who are into biotic stresses, is an important plant hormone in biotic stress resistance. It's also known that if you add ABA exogenously to rice plants, you'll get an increased in severity to disease for bacterial blight. So with those things in mind, we asked the relationship of high temperature ABA response, bacterial blight disease, and then we'll talk about the XA7 mediated resistance. So again, we're looking at the kernel density plot, and you can see the shift is again to the positive side. So we're seeing an upregulation of ABA biosynthesis and ABA responsive genes at high relative to low temperature in the disease interaction. And that's what we would have predicted from the literature. But now, if we ask the question, what happens if you put XA7 in this mix and the effector gene ABRXA7? And what you see here is that you have a dramatic downregulation in ABA responsive genes and biosynthetic genes. So XA7 is somehow blocking the upregulation of the ABA biosynthetic genes. So we also looked at other hormone responses because they can be interesting. And in fact, as you might expect, the salicylic acid pathway is shifted up to a certain extent in the disease interaction. This is not a resistant interaction in the black line. But with XA7, it's downregulated, which is not what we would have expected at high relative to low temperatures. And so we're wondering if there's something really unusual about the XA7 resistance gene and it's not using the SA pathway. So those are things that we really want to look at more as we move forward. So I want to kind of bring this around to what's known in other systems. So the only other system that's really been studied to any large extent for bacterial diseases is the Pseudomonas syringae interactions with Arabidopsis. And first you need to remember that Pseudomonas syringae Arabidopsis is a temperate disease, not a tropical disease. So we're talking about two different pathosystems. It's an between the cell type pathogens, whereas Xanthomonas oryzae is a vascular pathogen. So we're talking about very different pathogen systems, monocot versus dicot. So a little bit different, but I think we can learn lessons by comparing these together. So at high temperatures, they see an increase in Pseudomonas syringae replication. And we saw that to a certain extent, but it was not, clearly not the mediating factor for bacterial blight. We don't know yet if we have an increase in the effector translocation, the movement of the AVRXA7 gene from the bacteria into the plant. They've shown that for Pseudomonas syringae. And they see a decrease in the production of a toxin called caronitin, which for that bacteria is an important virulence factor. In fact, it makes the stomates open so the bacteria can get in. So for them, that's an important factor. For us, it's not an important factor. They see an increase in ABA in the disease-susceptible interactions, which we also saw. But remember, with effector gene AVRXA7 present, we saw the downregulation. They see a decrease in SA in the susceptible interactions. And in fact, we saw a decrease in the SA production. They see decreased effector-triggered immunity. That's disease resistance that's driven by single genes. We haven't measured the other resistance genes. We're in the process of doing that now. But with XA7, we see the opposite. We see an increase in effector-triggered immunity with XA7. And we have not measured PTI, PAMP-triggered immunity, or the basal resistance with this interaction, but they see an increase at high temperature. So what we know is that there are a lot of interesting things going on across different pathogens. So one story isn't going to be the story for all, but we also know that there are some unique things about interactions in rice, particularly with the XA7 gene, that make that story very interesting. So why are we interested in this? So just to summarize, we know that the ABA biosynthetic and responsive genes are upregulated by both heat and combined heat and disease stresses, just like Stephen measured in the core responses. But they're suppressed by XA7-mediated resistance when we have high temperatures. So our hypotheses moving forward are that during simultaneous high temperature, stress, and pathogen attack, rice favors the abiotic stress pathway. So if we have both high temperature and disease, we're going to favor this ABA pathway. But if you introduce XA7 in, XA7 is more effective because it suppresses the abiotic stress response pathway. Now, what are the implications of this, and why do we care? Well, in the long term, we want to be able to identify sources of resistance that are effective at high temperature and don't break apart, that don't fail at high temperature, and to facilitate crop breeding into the future. And what I tell my students is the key thing is we want to avoid mistakes. We want to make sure that if we breed for high temperature, we're not putting a gene in there that's going to break down when we put it out in the field with disease. And I can tell you from personal observation that there are examples where the breeders of rice have worked hard to get a resistance to drought stress tolerance into rice, only to put it in the field and have it break down due to pathogen pressure. So we need to think about how do we breed for multiple stresses, because plants don't stand out in a field and they're not affected by only one stress. And that brings me to, this system is complex. The phytobiome is complex. And so in the changing climate, we not only need to think about the plant pathogen interactions, which we have done for years, but we need to think about them in the context of the environment in which the plant is hosted and the other stresses that that plant is under out in that field. So with that, I'll point out, so when you're as old as I am, you have a long career of students and collaborators. And the students that have been involved in this, it started with Kim Webb, Stephen Cohen, and Nali Vera Cruz, and Hong Xia. Jen Ship is in the lab now, and she's taken over the project with the help of a bioinformaticist, Alvaro Perez, who's now back here in France. And then thanking my funding and the other lab members. So thank you very much, and I'd be happy to take any questions. Yeah.
Some questions?
Mm-hmm.
XA7 is executor protein.
It's a
XA7 is executor protein.
Yes.
It's a protein normally localized in the chloroplast, if I remember. No,
I think you're correct. I think you're correct. Yeah, it's a small protein of unknown function. Yeah, yeah.
And did you manage the signaling pathway between the executor protein function, the photosynthesis regulation, and the ABA pathway, or this is something?
We know that photosynthesis is downregulated in these interactions, and that's about, we haven't, but that's a good point. We haven't tried to make that connection to see, yeah, that's a good point. We really haven't, because XA7 was just cloned late last year, early this year, so we're still getting our hands and heads around it. Yeah, that's a good question. Yeah.
Maybe we can discuss later. I have some tracks about this protein.
Ah, oh, great.
But in Arabidopsis, not in rice.
Yeah, that's okay. We can translate it to rice.
I take over. Thank you for the talk. With respect to this gene being upregulated at high temperature, my first question would be how many other genes follow the same pattern? And then what could be our thoughts about such genes being there in rice? And as in the rice history, something happened to basically respond to such stress.
Mm-hmm.
And then with respect to the interaction increasing maybe in the promoter region, can we just imagine that this is like in thermophilic bacteria, some kind of interactions being stronger because of the structure of the DNA and something happening around these lines?
Mm-hmm. So the first question was, are there other R genes that we see that have that effect of XA7? We've looked at probably of the 30 genes out there, not all have been cloned, of course, but that doesn't matter. We can ask the question. We've looked at probably 10 different genes and have never seen an effect like we see with XA7. They all are less effective at high temperature, which is particularly alarming to us. I also didn't say, and I should have said, that the genetic background of the host can make a difference. It can modify that to a certain extent. So XA4 doesn't break down in India where it's 40 degrees. It's somewhat effective, but it's less effective. The second question was the evolution of the genes, or
if there are many genes, even if they are not resistant genes, but if there are many genes being upregulated by high temperature in rice, where could that come from?
So XA7 comes, the history of XA7 is from a variety of rice that is from a high temperature environment and from also a drought stress tolerant variety. So it could have evolved under those settings, and that could be how it evolved to change that way. What we don't know is if, because we don't understand truly, but my guess is there's something to do with the structure of the DNA at the high temperatures or some effect of that. What we're interested in from a broader perspective in my group is the relationship of promoters across the genome in defense responsive genes. So we study basal resistance and the regulation of genes involved in basal resistance. So the genes that as I call them, the generals, the R genes tell to turn on. Our hypothesis is if those are better regulated, they get upregulated faster or more efficiently, we could have better resistance, because we know they're present in resistant and susceptible varieties. They're just not well activated between the two. So we've been studying the promoter regions and the clusters of regulatory elements in those promoter regions, some of which are the effector binding elements that the tau effectors regulate. But these clusters of cis-regulatory elements respond to fungi, bacteria, anything that activates a stress response in the host. So the evolution of those and the accumulation of those has partially been driven by the accumulation of quantitative resistance accidentally in breeding programs. But also, if we look at where the sources of those good promoters are, it's frequently in rice varieties that are from, the rice variety I'm thinking of that we can get several of them from is a drought stress and disease tolerant variety. So I think in nature, they have accumulated accidentally. And then in the breeding programs, we've separated them because we've been breeding for single gene resistance and not really paying attention to the other things. But now if we have a mechanism to target them, we can bring them in as well. That was a long-winded answer, but did it answer your question?
I asked too many questions.
It's okay.
There are some questions in the chat, so I'm going just to tell you the question. So a question from Dominique. Do we know in which cell this sugar transporter express? Does it have a role on osmotic regulation and also in stomata closure?
So we know that they can be, for example, Xanthomonas oryzae is a vascular pathogen. So we know the xylem parenchyma can express those sugar transporter genes, and we know that they can be expressed in the parenchyma cells of a leaf, because we know, for example, that some of the xanthomonids are not vascular pathogens, and we can see the genes expressed. And actually, there are some new tags that allow you to watch the expression of those genes. What I don't know, because Wolf Fromer is an expert in this field, and if I would target you to his papers, I don't know much about the osmoticum change with those transporters. I don't know if the amount of sugar that they secrete is significant enough to change the osmoticum of the environment. But I would tell you to talk to Wolf Fromer, because that's his field of expertise, and he studies these things.
So another question in the room, or they are over in the chat? So a question from Matthias. C'est bon, c'est bon. C'est bon, c'est bon, c'est bon.
Work. Do we have other example of study of the effect of global warming on other pathosystems, such as fungal diseases? Do you have some examples? Is there a comparison between bacterial diseases, fungal disease?
So there are studies, especially now, a lot of studies are emerging And the story is not going to be the same. So for some fungal diseases, for example, high temperatures, the disease is less. And I suspect it has to do with probably when they're growing along the surface of the leaf and things, once they're in the plant, who knows? But the literature to date suggests that in some cases it's lower. In fact, there's literature out there on magnaporthy, and I think that most of the R genes are not as effective at high temperature, but I don't know how far that's been looked at. There are examples in wheat, for example. In fact, one of the most well-studied cases is in wheat. And there's a resistance gene to a rust fungus that is effective at high temperature. I think I've got that right. Yeah, I haven't thought about it for a while. Yeah, but there are differences. It's not going to be the same. So some cases you're going to see the plants, you move them into a new environment or the temperature goes up, they're going to be fine. They're going to be better off. But then, according to that study that came out recently in Nature Climate, the new pathogens will evolve, and so we will, as we move the plants into new environments, and new pathogens can adapt to those plants. So I think we need to think about very carefully what we're going to face in the future. That was a rambling answer, but...
And another question from the chat, from Cecile. Do we know if rice plants expressing XA7 are more resistant or more sensitive to drought or cold stress?
That's a really good question, and we have not asked that question. Again, you have to have the effector there for the XA7 gene to be expressed. So you have to have the pathogen present in order for XA7 to be expressed. So you would have to make those studies under a disease situation, and we haven't done that. But that would be the caveat to that kind of a study. Yeah.
There is still one question in the chat, so I'm going to ask it. So from Bernd, who say thanks a lot for your great talk. First, he said, may I ask you if you have any information on fungal diseases on temperature impact?
Yes, and I think the best literature out there is in the wheat systems, so I would point you to that. And other diseases, in some cases, as I mentioned earlier, the plants are more resistant at high temperature to fungal infection than at low temperature, and I suspect it's to do with the fungi themselves. But I think we don't know it for very many cases. Only a few cases have been studied well, and I think they're the rust pathogens and also rice blast.
Yeah. I wonder about the jasmonic acid pathway and the balance, of course, between salicylic acid and jasmonic acid pathway, the hormonal, but also the interaction with the abscisic acids and jasmonic acids in heat shock, heat stress.
Yeah. We looked at the jasmonic acid regulation in this case, and I don't remember seeing anything that stood out to us. In general, the relationship between jasmonate and ABA, I don't know. We work with we do work with an aphid and bacteria associated with aphids, and in that case, the jasmonate pathway, once it's activated in insect interactions, is a defense response. But bacteria associated with the aphid suppress by inducing an SA response, they suppress a JA response. But I don't know what happens to ABA in those cases. I don't think we've looked. It's a good question. Yeah. Yeah, the hormone, as you're well aware, the hormone interactions are very complex in plants, and yeah. Yeah, that's a very good question. I don't know the answer to that.
Just both of them close the stomata, abscisic acid and jasmonic acid.
Yes, yes, yeah. In dicots, for sure, they close the stomata. And in rice, I know you can get stomatal closure with the JA story, but for this pathogen, which is it goes in through the hydathodes, it doesn't go in through the stomates. It's not quite as well studied a story, so I guess I don't know the answer to that, but yeah. Yeah.
Me again, if I may.
Mm-hmm.
I caught your comment at the beginning about moving crops somewhere and thinking it will do the job, and there are so many different aspects with respect to soil nutrition and so on. In this case of resistance that you showed, have you looked at what could happen in the plant with respect to resources and changes in metabolism that would participate in the fact that the crop is being resistant? And does that open a door for the future of thinking how to help the breeders, maybe with the nutrition of the plant and not only with the plant itself?
It's a great idea, but we have not even touched that. I mean, we have the transcriptome data where we could go in and look at different nutritional pathways and what's changed. We haven't looked at that. And to my knowledge, I mean, that's part of that whole system that we need to be thinking broader in. We need to be thinking of it. And I don't know that other I know, you know, we talk about helping, for example, with citrus greening disease by improving the nutrition of the plants to reduce disease. We don't do that so much in annual crops, do we? And perhaps maybe we should think a little bit more about that. Yeah, it's a good thought. Okay. Yeah, thank you.
There is still one question from Marie-Pascale.
A last question.
Yeah, I was surprised.
Marie-Pascale.
A question from Marie-Pascale was surprising. Do you have any information on various diseases versus temperature impact? So virus diseases versus temperature impact.
Disease temperature impacts? Oh, virus. Yeah. So there is an example of a virus temperature responsive gene, and it's very well studied as well, and I'm trying to think of who and what. And in that particular system, the resistance breaks down at high temperature, I'm pretty sure. But I don't know beyond that. Yeah. Yeah. And I'm probably not up to date in the virus literature, I can tell you.
Okay. Thank you, everybody.
Thank you.